You meet someone at a party. You feel a first spark, an initial connection. Is that enough for a true bond to form and last? Probably not. That's the same problem cell engager therapies face. These therapies are designed to help the immune system detect and kill target cells. And the gold standard for selecting the best candidates for these therapies is avidity, how strongly two molecules bind. But that's only part of the story. What truly matters is not just the initial binding, but how their interaction remains stable under real dynamic conditions, capturing all the bonds that anchor them to each other, showing the full story. That's avidity. And it is what truly predicts whether immune cells will stay attached long and strong enough to kill target cells. At Avicell, we measure exactly that. We have built a microfluidic platform that mimics the human body, tracking which bispecific antibodies and immune cells stay attached to target cells under increasing shear stress, allowing us to detect the best candidates, the ones that are going to work in vivo, in clinical trials, and ultimately in patients with 2 times higher approval odds. Because in cell engager therapies, it's not about what binds first, it's about what holds on long enough to kill. We are raising £2 million to complete design validation and in vitro testing. Join us in transforming how therapies are selected. Avicell, smarter decisions, better therapies.
Can you share who will be your customers? Who do you want to sell to and to work with?
Yes, of course. So we had 2 choices. We could work as a CRO, or we can sell the actual devices. So we chose to follow the selling the actual devices path. So our main customers are going to be biopharmaceutical companies, research institutions, and CROs who need this platform to test cell engager therapies. So the main biopharmaceutical companies and research institutions that we're targeting are the ones that have this type of therapies in their pipeline.
So clarification, you talk about shear stress avidity. What's the difference between that and just plain old avidity, just for my information?
Yes, of course. So to answer that question, we need to have like the 3 different measurements in mind. So affinity is just how the 2 of them bind. Yeah. Avidity is how they form the synapse, where you see the different bonds that anchor them to each other. But then when you add the shear stress, you have functional avidity. So it actually measures how all of these bonds actually interact with the shear stress force and how they behave and if they remain attached or not. So it's just like increasing them and going more in depth about how the actual binding is working between the cells.
Okay. So if a pharma company or a CRO was to bring this into their discovery group, where in the workflow do you see this being used?
Yes, of course. It is going to be in the in vitro phase of the drug development process. So when they are analyzing the different molecules and when they still have various like candidates, for example, for bispecific antibodies, and they have different options, and they want to figure out which one is the most, like the better one, and the one that it's going to work further along the path. This platform fits there to help them reduce the amount of candidates and select the best ones to continue during the development process.